This UCSD TV program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest programs.
Good evening and welcome to the Jeffrey B. Graham Perspectives on Ocean Science Speaker Series. My name is Cheryl Peach and I'm a program scientist here at the Birch Aquarium at Scripps Institution of Oceanography. It's my great pleasure this evening to introduce our speaker, Dr. Jen Smith. Jen is a coral reef ecologist and an associate professor here at Scripps. She received her PhD from the University of Hawaii, where she remained for several years as a faculty researcher, and then she came to Scripps as a postdoctoral scholar, and fortunately for us, decided to stay on and is now a valued member of the faculty here. Jen's primary research interests are actually in marine ecology and in understanding how various physical and biological processes affect things like coral reef communities. Jen does a lot of work on stressors that might affect such communities, everything from local effects like pollution or overfishing to more global effects like our warming oceans and ocean acidification. And she's going to share some of her research with us tonight. So please welcome Jen for her talk titled Coral Reef Ecosystems, Human Impacts, Pristine Reefs, and Conservation Strategies. Jen, thank you so much for being here.
Thank you, Cheryl, for that lovely introduction. It's a pleasure to be here tonight to share with all of you some of the research that I do down the hill at Scripps. As Cheryl said, I'm a marine ecologist, and my research interests are really in understanding the factors that structure different marine ecosystems. And I'm primarily focused on shallow water marine ecosystems, those that you can visit by scuba diving. So we're not talking about things in deep water that we visit in big ships, such as the Scripps fleet, but we're literally scuba diving and collecting our data underwater firsthand. So I'm going to focus tonight on coral reef ecosystems, some of the work that we do in the tropics. And for any of you who have been fortunate enough to scuba dive or snorkel on coral reefs, you'll share in my enthusiasm for these spectacular ecosystems. They're some of the most beautiful, brilliant, colorful, complex ecosystems that support many species of charismatic fauna, such as this vampire snapper. They support many different species of fish and vertebrates and plants. They can be home to many, many individuals of species, such as that convict tang. The corals create these complex landscapes that provide habitat and shelter for many other organisms. And they're just these spectacularly beautiful ecosystems that are wonderful to visit. And that's why many of us just share an innate passion for these ecosystems. They're also important for a whole variety of reasons. Coral reefs are believed to be the most biodiverse ecosystem in the ocean. So even though coral reefs occupy an area of the planet smaller than the country of France, they house more species than any other ecosystem in the ocean. So biodiversity, anywhere between 1 and 9 million species are estimated to live on a coral reef. We don't really have a good idea of how many species actually live there. They're also really important for providing physical protection. So this is a coral reef. This is a living geologic feature that you can see from space. The coral organisms are growing up towards the sun, creating this massive barrier around this tropical island. And this is created by living organisms. And this provides a huge ecosystem service for tropical islands, particularly in the form of protecting them from the physical erosive forces of wave energy. We know that tropical locations get many tsunamis, they get tropical storms, hurricanes, cyclones, and just large swell such as what is on its way to Hawaii right now from Alaska. Many of you have heard of this mega superstorm that's in Alaska, sending waves on the order of 25 feet high to the north shore of the Hawaiian Islands. So these living reefs provide coastal protection, preventing those islands from eroding into the sea. They're also really important for tourism. Many of us are willing to pay money to go visit coral reefs, to interact with them, to scuba dive, and to experience that natural beauty that they provide. They're also important for fisheries. Many countries that live in the tropics or that exist in the tropics, the people of those countries depend primarily on coral reef fishes, not things like tuna and big pelagic fish like marlin and jacks, but things like reef fish such as parrotfish and grouper, snapper, surgeonfish, fish that actually live on the coral reef itself. So many of these people depend on these fish for their primary source of protein. And then of course reefs are also important for their natural beauty, as I've mentioned. And these pictures, while familiar to me and are kind of the iconic photographic sequence of a healthy coral reef, have become less and less common on reefs around the world, and I'll mention that briefly in a minute. So this is all of these ecosystem services that I mentioned are all due to the life of these coral organisms. So this is a single coral individual, a coral polyp. It looks just like a sea anemone. They're very simple little organisms, and corals actually form colonies. So this little polyp will bud to form many copies of itself. So this is a colonial coral with lots of little polyps that are all connected together. Their stomachs are all connected and they share resources, and they're all the same genetic entity. And these corals can continue to grow, creating clones of one another, in eventually forming some of the largest living organisms that we have on the planet. So this is one living coral. It's about 30 feet across and about 20 feet tall, and it's probably over 500 years old. We can tell by drilling cores into these corals, and they leave behind annual growth rings that are similar to how we age trees. So corals are these living geologic features, as I mentioned, and as that small little coral polyp grows, it's leaving behind a calcium carbonate skeleton underneath it. So when we think about this organism, this is all stone, it's all calcium carbonate, and only the yellow part on the top is the living tissue with the fine little fleshy coral polyps. So as it grows, it's leaving behind this calcium carbonate skeleton, creating those living reefs, the massive geologic structures, and eventually creating these complex landscapes. So all of these are stony corals. They're hard corals. They're basically rock, but they're living with just that fine veneer of tissue over the top. And we get corals that branch, that form staghorn or antler corals. We get cauliflower corals. We get these massive or mounding corals, plating corals, encrusting corals. And all of this creates that complex heterogeneous landscape that provides a lot of different types of habitat for all the species that live on the reef. However, many of you are also probably familiar that coral reefs are undergoing a state of global crisis. It's estimated that 20% of the world's reefs have been lost. Another 25% are in imminent danger, and this is largely due to anthropogenic or human-caused impacts. These impacts occur at both global and local scales, as Cheryl mentioned. And what I mean when I say we're losing coral reefs is that we see a coral-dominated landscape, such as seen here, undergo what's known as a phase shift to dominance by these fleshy seaweeds. So these are all algae or seaweeds, primary producers that are faster growing, opportunistic. They can colonize empty space really quickly. And so those corals are really fragile, and when they are suffering impacts from a variety of different sources that I'll mention, those fleshy seaweeds can overgrow them. So this is a reef in Maui that I've been working at for the last 15 or more years. And I've watched that reef get overgrown by all of this fuzzy, fleshy macroalgae, which is, you can see it's basically choking that coral out. And the corals are very slow growing. They can live for hundreds, thousands of years. And so when they get overgrown and choked out by these seaweeds, it's pretty much the end of the road for that reef. We've seen this happen on reefs around the world, and probably the most well-known is what's happened throughout the entire Caribbean, where these staghorn or antler coral, the acropora, used to dominate throughout much of the Caribbean, even places like Jamaica, and now much of the Caribbean is covered with these different fleshy seaweeds. And so this has been a phenomenon that we've seen around the world. These phase shifts from coral to macroalgal dominance, and in the worst case scenario, we call this the slippery slope to slime. So in this case, this reef is now covered in slime, and once a reef has transitioned into these states of seaweed dominance, it's really hard to get them back out. So that is what my lab at Scripps focuses on, this fun group of lovely students, master's students, undergraduates, PhD students, and postdocs. We all are interested in determining the effects of various anthropogenic or human-derived disturbances or impacts, how humans impact coral reefs, looking at both global and local stressors. We're also interested in understanding what coral reefs look like in the absence of people. And so in order to properly manage coral reefs, we need to know what they looked like before humans started messing with them. And so to do this, we go to pretty much the most remote parts of the planet to try to see what coral reefs look like in the absence of humans. And then both of these different research areas are ultimately tied together to try to find ways of developing better restoration and management strategies to protect and preserve these ecosystems for future generations. So we'll start with the first one. When we think about those corals going away, there are two kind of main mechanisms by which they can go away. They can be killed directly, and these types of large-scale coral mortality events usually occur on larger global scales. So many of you have heard of how global warming causes coral bleaching. When the water gets too warm for too long, the coral has to release its symbiotic little algae that live inside of its tissue that provide it the primary food source. It releases those algae, and eventually the coral suffers and will eventually die. And this has been kind of one of the largest causes of massive coral mortality across much of the Pacific. Ocean acidification is a new problem, or perhaps not new, but something that's a new area of research in terms of looking at how it impacts coral health, and is an area that I'll mention more about. Coral disease, we've seen disease outbreaks that have swept through places like the Caribbean and killed a lot of coral. And things like storm damage, big hurricanes can knock out and wipe out large areas of coral reef. So I'm going to focus, we can spend a night literally talking about each one of these. I'm going to talk briefly about ocean acidification, because this is an area that's actively being studied and is something that's certainly timely. So ocean acidification occurs as the result of burning of fossil fuels, which causes carbon dioxide to increase in our atmosphere, while simultaneously cutting down massive areas of forest. So trees absorb carbon dioxide and use it for photosynthesis and turn it into tissue, and burning a fossil fuel releases carbon dioxide. So doing these two things simultaneously really increases the amount of CO2 that we have in our atmosphere. We know CO2 has been increasing in our atmosphere thanks to Charles Keeling, who was here at UCSD. Keeling started measuring CO2 in the atmosphere in the 50s, in the 1950s, so starting here. But prior to that, we have data using air bubbles trapped in ice, using large ice cores. And you can basically see that over time, since we've been able to detect this, we've seen a massive increase in CO2 in the atmosphere. In fact, we've never seen CO2 levels we haven't seen CO2 levels this high since humans were on the planet. I think for the last something like 800,000 years, we haven't seen CO2 levels this high, and we've never seen a rate of CO2 increase at the rate that we're currently seeing. So CO2 is increasing in the atmosphere. We know that. The red line shows CO2 in the atmosphere. From stations monitoring the open ocean, we also know that CO2 is increasing in the surface waters of the ocean. So this is taken from ships that go out and measure CO2 concentrations in the open ocean, and simultaneously with that rise in CO2, we get a decrease in pH. So this is what's known as ocean acidification. So the pH of the ocean is slowly decreasing, and why does that matter? This shows how the chemistry in the ocean changes as CO2 dissolves. It undergoes a series of chemical reactions, ultimately leading to more bicarbonate and more hydrogen ion and less of this carbonate. So we get a reduction in pH, an increase in PCO2, or the amount of dissolved CO2 in the water, and a decrease in omega, which is the amount of carbonate that's in the water. And this is important because all of those organisms that build shells and skeletons need carbonate to make their calcium carbonate. So that's the primary building block. So we're not only reducing pH, but we're reducing the amount of that particular ion that's available. So this is problematic, and we are interested in my lab at looking at the effects of ocean acidification on key species. So how does OA, or how will ocean acidification likely affect certain species that we may care about? Things like commercially important species like oysters, or things like corals, which we know build their shells and skeletons out of that calcium carbonate. We also are interested in looking at the evidence for ocean acidification in shallow nearshore ecosystems, because all of the measurements that have occurred so far have been in the open ocean very far away from any coastlines. So not in any place where we have a kelp forest or a coral reef. And then lastly, we're interested in using some natural biological processes to potentially mitigate ocean acidification. So I'll start with the first, looking at the effects of OA on key species. And to do that, we use experiments. So in my lab at Scripps, we have a little experimental aquarium where we can bubble in carbon dioxide into little experimental mesocosms, and we put species of invertebrates or different types of seaweed in those jars, and we measure the responses of their physiology to increases in CO2. We're particularly interested in looking at some of those seaweeds. So why is that? Many fleshy seaweeds, such as these species shown here, they're going to be able to use elevated CO2 because they're photosynthetic. So the primary substrate for photosynthesis is CO2. So they fix CO2 and they release oxygen and they turn that into tissue. So elevated CO2 could potentially increase the productivity of these different fleshy seaweeds. Whereas we have many different calcified species of seaweeds. These are all very important in the tropics. Some help build those reefs that I mentioned. And we could predict that CO2 might also increase their productivity because they're also photosynthetic. But because we have a decrease in the saturation state of calcium carbonate, we might see a reduction in their calcification. So we wanted to test this to see if elevated CO2 would increase these guys while decreasing these species. And so we set up an experiment, or several experiments, working in the remote island of Palmyra, which is located in the middle of the Central Pacific. This is an atoll that Scripps, as a Scripps scientist, we're members of the Palmyra Atoll Research Consortium. This is a wonderful study location because it's very far removed from any local human populations. And we have a little tiny field station on the island in conjunction with the Nature Conservancy. Palmyra used to be occupied during World War II, so it has a landing strip conveniently that has been upgraded so we can fly into the island and spend several weeks working there. And the reefs are actually still in quite good shape, given that there's no local human activity there. So one of my graduate students, Maggie Johnson, has done a whole series of experiments with all of these different species that I mentioned and measuring a variety of different responses just by simply bubbling carbon dioxide into these little aquaria and measuring the responses of those species. So I don't want you guys to really absorb everything in this graph, but the important part is these are the fleshy species that we expected would respond positively to ocean acidification, and all of the red dots are the CO2 treatment, and you'll just notice that the red symbols are typically higher than the blue symbols for those fleshy species. The calcified species are shown over here in gray, and we expected them to respond negatively to CO2. And you'll notice that over here, the red symbols are all lower than the blue symbols. If we put all this together in a more digestible graph, you basically see that if we combine all those experiments together, the fleshy species are enhanced by elevated CO2, while the calcified species are negatively affected by elevated CO2. So this is the result of actually several experiments, about 16 different species that we studied. And ultimately, we've been able to show that high CO2 does enhance the fleshy species while reducing the growth of those calcified species. And so while we still have a lot more work to do, there are hundreds, thousands of species that live on a coral reef, our data so far suggests that the corrosive impacts of ocean acidification is likely to harm the abundance of these important reef builders, the calcium carbonate depositors. There's been numerous studies on the reef building corals themselves, and pretty much, well, there's a lot of variable results, but a lot of results have also shown negative effects on the corals as well. While elevated CO2 will also enhance the abundance of these fleshy species. So this is evidence kind of in support of how those phase shifts might happen in the future with elevated CO2. We're fertilizing these fleshy species with carbon dioxide while eroding the calcium carbonate builders, the reef builders. So this has important implications, obviously, for the future persistence of reefs. But what is actually happening in the seawater on these reefs? How is pH varying? Are we seeing ocean acidification in action? And to understand this, we need to do monitoring. So as I mentioned, most ocean acidification monitoring data, long-term monitoring data, is from the open ocean, very far away from where things like corals or kelp or anything like that lives. And so we need to monitor ocean acidification in these shallow coastal systems to see if we can measure this OA, ocean acidification, in action. And so to do this, we have an active monitoring platform off the Scripps Pier. My lab's been trying to maintain this on a shoestring budget for the last several years. You'd think it would be something that would be institutionalized. We're hoping to get that to happen in the future. But so we're monitoring ocean acidification here right off the coast, and then we also have a program in Palmyra, the remote island that I mentioned, where we install these sensors. So specifically these CFET sensors, which were designed by a professor here at Scripps, who's a close collaborator of mine, Todd Martz. With the development of these sensors, we're finally able to monitor pH autonomously without having to take water samples every time. We basically can program these sensors and deploy them and leave them in the field for months at a time, pick them up, download them, and have continuous time series data of pH. And this has only been possible really for the last few years. So thanks to Todd, we've been able to develop data sets like this. And this is the results of several studies across multiple ecosystems around the world, but the point being that if we look at the two areas in red, the open ocean and coral reef ecosystems where some of my data sets contributed, we see that the open ocean is basically looks like a flat line. And then if we were to, this is only a 30-day time series, if we were to carry this out for years on end, we would expect these lines to gradually be declining over time as ocean acidification happens. But we look at a coral reef and there's a lot more action going on here. There's what we call diurnal or daily variability. So each one of those bumps or humps is associated with a day light cycle. So that's interesting and something that we didn't have a whole lot of data on before, primarily because these sensors were only recently available. And we see this spatial and temporal variability on coral reefs, so that means over space and across time. And we see this really strong, as I said, diurnal or daily variability in pH, where during the day we get big spikes in pH, and at night we get these reductions in pH. That's called diurnal cycling. And an interesting thing to note is that every night the pH on these reefs is actually dropping below the values that are predicted to occur in 2100 as a result of ocean acidification. So this proposes all kinds of interesting hypotheses. You know, maybe these ecosystems are going to be more acclimated to reductions in pH because they're seeing these low pH values every night. But as the open ocean water continues to decrease, these values might still eventually decline and cause significant impacts. If we look at our data from across different sites, we see that in shallow protected environments like big lagoons or swimming, almost like a swimming pool, a big tidal pool, we get really high variation, whereas on the deeper four-reef communities where you have a lot of current and flushing, we have much lower variability. And so this suggests that as the parcel of water is sitting over a certain area of reef, the longer it stays there, the more variability you're going to have. Whereas if you're getting flushed with the open ocean, that variability is going to get swept away. So what is that attributed to? If we take a look again at these diurnal or daily fluctuations in pH, as I mentioned, we see big peaks in pH during the day and drops in pH at night. Peaks during the day, drops at night. And we see this pretty continuously over time. And so what is causing that? Well, it turns out that the reef itself is causing that, right? So all the things that live on the bottom, many of the species that live on a coral reef are photosynthetic. So they're going to be taking up CO2 all day long, which is going to raise the pH, and they're going to be releasing oxygen as a byproduct of photosynthesis. And then at night, everything is respiring or exhaling, releasing carbon dioxide back into the environment, which reduces pH, and of course they're consuming oxygen as they're respiring as well. And so the whole process of the reef breathing and exhaling, inhaling and exhaling, photosynthesizing and respiring is causing those diurnal or daily variability, the daily peaks and troughs in pH. And so now we know that pH in shallow coastal ecosystems is much more variable than the open ocean, and we need to take that into account. We see this day-night cycling that's predictable and associated with the organisms that are living on the bottom. We also know that some species have much larger effects on that variability than others, and we're interested in learning whether we can use this information for mitigation. So that's the last module in this ocean acidification section. So we know that those fleshy seaweeds I talked about first, they respond positively to CO2 enrichment. And they have really high rates of photosynthesis. So they take up CO2, they increase pH due to those high rates of photosynthesis. And so it's been proposed that fleshy seaweeds might be able to alter the water chemistry around them to make it more favorable for calcification for species that are more sensitive, like the corals, like things here in California, like things we care about, such as oysters and abalone, the species that are commercially important. So might we be able to make a curtain of kelp around an oyster farm to protect that farm from reductions in pH occurring as a result of ocean acidification in the open ocean? On coral reefs, might we be able to use seaweed farms to help allow us to grow coral fragments for restoration projects? So this is an area of research that is very new, and there's a lot of proposals and hypotheses out in the scientific community, including several of my own. So I don't have any data to present on it, but it's certainly an exciting area of future research endeavor that hopefully I will have data on in the years to come. But this just shows an example. So in the tropics, there are many areas that grow large amounts of these fleshy seaweeds for a variety of different commercial production companies, such as the production of carrageenan, which is a cell wall product in seaweeds that are used in things like shaving cream, whipped cream, sour cream, ice cream, toothpaste, a whole variety of human products. And so these are massive farms that occur over large scales. And so how are these farms affecting seawater chemistry, and might we be able to use them to our favor to help us mitigate ocean acidification? It's been proposed that kelp could be the seaweed that could save mankind, and I would say, well, yeah, but can it? So the question is still out, the jury's still out, and we've got a lot of work to do to try to answer that question. Okay, I'm going to move now to talking about some of the local causes of coral reef loss. So we talked about ocean acidification as being one of these global issues, and it's something that really needs to be managed on a global level. CO2 is a global problem. However, there are plenty of things that occur on a local scale, things that occur more on in our backyard kind of scale, where managing them is a lot more easy. Things such as overfishing, pollution, sedimentation associated with deforestation, the introduction of non-native species. So all of these things occur on smaller scales, scales that are much more easily manageable and manageable in our local communities. So on coral reefs, overfishing is a big problem. There's a variety of different ways that people fish destructively on coral reefs. Dynamite fishing, cyanide fishing, the use of gill nets, all these things not only extract fish, but can also harm the habitat. Also, coral reef fisheries tend to be somewhat non-selective in that some of these techniques like gill netting is not targeting just one species. It's basically catching anything that swims into that net. And so along with that, we end up killing a lot of species of fish that are really important for maintaining a healthy reef. And in my lab, we're particularly interested in the herbivores. Those are the species of fish that eat the seaweeds that overgrow the corals that we care so much about. So on a coral reef, you have an abundant and diverse population of herbivorous fish, as shown in this video. These are parrotfish and surgeonfish, which are actively mowing the lawn, right? You can see them taking, try to count how many bites they're taking per minute. That they do this all day long. They swim around the reef, and they're eating all these little seaweeds that are growing on this bare carbonate, which allows the seaweeds to stay in very low abundance, which prevents those seaweeds from overgrowing and killing the coral that are much more slow growing. So these herbivores, the herbivorous fish, are very important to have around. We've done a number of experiments around the world where you basically put up little cages to remove your herbivores and see how the bottom of the reef changes over time. We've done these experiments in a whole variety of different locations, and time again we found that when you remove herbivores, you get more herb or more algae, more seaweeds, and less coral. And so it's not rocket science, but in the scientific community, you have to prove that these things are important before everyone will believe you. So we know that these herbivores are very important for maintaining a balance of coral to algae on a reef. And so we've been moving to try to promote through all of the research we've done over the years to try to promote the development of herbivore restoration areas. So I'm sure all of you have heard about marine protected areas. This is usually no-take areas where we say we're going to set aside this chunk of reef and there's going to be no fishing allowed. Well, in a lot of the remote tropical places that we work, telling people, these local communities, that they can't fish anything is just not practical. But if we can focus in on a certain set of species that we know are very important for keeping the reef healthy, such as these parrotfish and other herbivores, it might be a more sustainable solution. So we've worked with the state of Hawaii to develop the world's first herbivore fisheries management area. This is at Kahekili Beach, or Airport Beach, off of the Ka'anapali coast on Maui. It was established in 2009 and has been a very great tool for educating the public about the importance of herbivores and for also promoting the abundance of all of these important species. Since the establishment of the protected area, we've seen an increase in the abundance of the herbivores themselves, and we've also seen the reef starting to respond on the bottom. So the reef is actually showing signs that it's becoming more healthy since the management area went into place. And it went into place in this particular reef because it was being overgrown with seaweed. So this is promising and suggests that this could be a useful tool for helping us not only maintain healthy reefs, but potentially to reverse the declining trajectory on a lot of reefs around the world. So we need more herbivore fisheries management areas, and my PhD student, Emily Kelly, has been actively working at this site for the last several years. However, this is still really the only specific herbivore fisheries management area in the world. And so my former master's student, Clint Edwards, and I were interested in asking the question, okay, well, what's the evidence that coral reef herbivores around the world are globally overfished? Are people, maybe we're just making a big deal of it, but it's really not that big of a problem. So we collected data, synthesized the literature, and collaborated with a bunch of scientists to develop this figure, where in blue, these are unfished areas, and this is the mass of herbivores on reefs all across the world, from the Caribbean, the Indian Ocean, and the Pacific. And you'll just note that there's a lot more big blue bars than there are red bars, right? And if we take a closer look at just the overall, the global averages of these things, the total amount of herbivore biomass, or grams of herbivore per meter squared, is more than twice as high in unfished areas than it is in fished areas. And we also see differences in the type of herbivore, with all of the important grazers that remove algae from the bottom declining in areas that are fished. Whereas we have this one group, territorial damselfish, which actually create little algal lawns. They create little seaweed farms, and they actively defend their territories from other fish. So the territorial damselfish actually promote seaweed. So with fishing, we're reducing all of the species that actually matter for removing algae off of reefs, and we're increasing the abundance of the species that create seaweed farms. So this synthesis was just published this year, and we're hoping this will provide incentive for other locations around the world to develop more of these herbivore protection areas, especially now that we know if you don't fish, this is how much biomass, or how many grams of herbivore you should have per meter squared at a global level. So we're providing kind of target values for places to know whether or not you're at a really low level. Is your reef characteristic of a really heavily overfished area, or are you doing pretty well in comparison to the rest of the world? Moving on from that, that's an area where we're trying to actively use our research to promote or help develop management strategies that could help reefs recover in the future. So moving on to human impacts and pollution, people provide a whole variety of different sources of pollutants into the water. For our purposes, thinking about the balance between seaweeds and corals, what we're particularly interested in is adding nitrogen and phosphorus to the water. So this is fertilizer. You think about what you put on your yard or your garden, you add fertilizer, which is nitrogen and phosphorus, and that makes your plants grow faster. Well, the same thing happens in the ocean. If you dump nitrogen and phosphorus onto a coral reef, your weeds will grow faster, and they will outgrow your coral. So how do we get nitrogen and phosphorus into the water? Sewage is probably the number one big impact. Runoff from rivers, especially when you have upland farming, agriculture, you have pig farms or cattle farms, all of that waste will eventually wash down into the marine environment, and that is basically nitrogen and phosphorus, which will fertilize your weeds. And so typically the reason why this is such a problem on coral reefs is because the water in the tropics on coral reefs is considered oligotrophic, and what that means is it's low in nutrients. It's not like our coast here where we have naturally high nutrients and we have kelp forest dominating. That's a completely natural process because we have upwelling occurring here that brings naturally high levels of nutrients. In coral reefs, the water is usually crystal clear. It's very low in nutrients, and that's why corals do so well in that environment. When we add a bunch of excess nutrients, nitrogen and phosphorus, we get our weeds growing. And again, we've done numerous experiments adding nitrogen and phosphorus and seeing how the seaweeds respond, and typically we've been able to link excess nutrients to these seaweed blooms in a lot of places around the world. Another specific case study is an area that I've been working for a long time, the island of Maui. And back in the early 2000s, we noticed that there were 3 areas along the coastline that had massive seaweed blooms, where these seaweeds would wash up on the shore and they would rot and create a foul odor, and it was causing big problems for Maui's economy. Tourists were unhappy with staying in certain hotels because it literally smelled so bad. And the reefs were getting coated in these different seaweeds, and was a big problem. So we noticed, okay, well, these blooms happen to coincidentally all be directly downstream from Maui's three large urban sewage treatment facilities. So is that a coincidence, or might there be some connection there? And the way that these sewage treatment facilities work is they settle out the waste and then they inject the settled out waste into the ground. And these are wells that are not capped with cement. They're literally just holes that are dug into the ground. Most of them are, the base of the well is near sea level. They're mostly all malca or up on the slope of the mountains a little bit, less than a mile from shore. And so we began wondering whether all of this waste was eventually percolating through the basalt or Swiss cheese-like rock that the island is made out of, and making its way onto the reef and causing these big algal blooms. So to start working with a former master's student of mine, we decided to see if we could use the tissue and the algae as a tracer for human waste. So there's something called nitrogen isotopes, and you can use the values of these nitrogen isotopes to indicate different types of sources of nutrients, of nitrogen. So we developed this map. We collected seaweeds from all over the entire coast of Maui, and basically higher values are more indicative of human-derived nitrogen than lower values. And you see the highest, the red dots here, those are our 3 places directly downstream from the sewage treatment facilities. So again, this is providing some evidence, but it's not a smoking gun. We were then able to obtain some of the injected effluent that they put into the injection wells, and we grew our blooming seaweeds with that injected wastewater. And you can see that as we increase in the concentration of the wastewater, we have happier and happier seaweeds. So we know that if you give the seaweeds this wastewater, they will positively respond, and they are, in fact, able to use the nutrients associated with that. And so this has been creating all kinds of contention among the Maui population and the government, and even at the time when I was still working in Hawaii or living in Hawaii, the government said, yeah, you know, we understand that this could be a problem, we just don't have the money to do anything about it. So, so that continued to go on, and the community continued to get upset. And then finally, the final smoking gun was a bunch of collaborators of mine were able to get permission to inject fluorescent dye into the injection wells on land and see if there were still plenty of critics that didn't believe that there was a connection between the wells and the reef. And so this was the final way that we were able to show that. And this is basically showing a little pipe that's stuck into the reef. And I'll just zoom up to here, but you can basically see green water coming out of that pipe, right? So this was 45 days after, sorry, the video is not that great. 45 days after the dye was injected into the wells, and we're seeing green water coming out. If it's even more impressive at night with a black light, then you can see it actually glowing. And so this was the final smoking gun that led a bunch of environmental nonprofits to sue the city and county of Maui over illegal discharge of wastewater. So this is basically in violation of the Clean Water Act. And I'm currently acting as an expert witness on this case. So this is a situation where all of this hard work and research that you've put in over the years will hopefully lead to a positive change in the community and lead to healthier water for not only Maui's reefs, but for everyone who cares about swimming and interacting in the ocean around the island of Maui. Okay, so now I'm just going to shift gears and briefly talk about hopefully that's given you a little bit of a taste for how we can do science that can actively be used for management, for solving problems on the ground to help increase the health of these coral reefs and hopefully help make sure that they persist into the future. But we also feel like we really need to understand what reefs look like in the absence of people, because most of the research that's been done on coral reefs began when scuba diving was first invented, and that was not that long ago. You know, only a handful of decades ago, maybe in the 40s or 50s. And so And actually, most of the reef research didn't really start until the 70s. And so what did coral reefs look like 200 years ago? You know, we have no idea. We have no way of going back in time. And the only way that we can really do this is to use a space for time comparison. We essentially can go back in a time machine by going to these remote locations in the middle of the Central Pacific that have no local human populations to see what reefs look like in the absence of human populations. And so this is really a collaborative effort between Dr. Forrest Rower at San Diego State, Dr. Stuart Sandin, who's in the back of the room, and myself, where we take our labs, we board ships, and we go into the middle of the Pacific to a series of islands that have that actually span a gradient of population density. This is the Line Islands archipelago, and the US has some jurisdiction over some islands, including Palmyra that I talked about, Kingman Reef, Jarvis, some of you may have heard of. And then the rest of the islands are under the jurisdiction of the Republic of Kiribati, which is an island nation that most people haven't heard about, but they have jurisdiction over 3 different island archipelagos, the Line Islands, the Gilbert Islands, and the Phoenix Islands. And some of the northern Line Islands are inhabited. The southern Line Islands, which include 5 islands, are all uninhabited, and they've never had local human populations. So we have a nice gradient of, you know, going from different levels of human population density. And we do these research cruises about every couple of years, where we go out there and this is really an opportunity for us to act like explorers. We get to go to places that no one's ever scuba dove on before. We get to collect data from places that potentially, or in many of these cases, no one's ever done science there before. And so it's a really neat opportunity, and is just great exploration. And once we're out there, we study everything from microbes and water chemistry to the benthos, the coral reef community, to the fish population. So we like to say from microbes to sharks and everything in between. And when we go to these places, especially the uninhabited islands, we find the water to be crystal clear, there to be very low nutrients and very little microbial abundance, certainly low pathogenic bacteria. You don't see any of those nasty bacteria that can cause diseases. We see a lot of corals, so the reef bottom is dominated by reef-building corals, the animals that we care so much about, and the fish populations are dominated by top predators. So this is what a reef in the absence of humans looks like. If we take a look at just the corals and we compare coral cover across the whole suite of islands that we work, this is 56 islands across the Central Pacific, including 5 different archipelagos. We first see that the inhabited islands, so where people are, there's just a lot lower coral cover, right, than places where we have uninhabited islands. And so that's, you know, essentially we already knew that coral cover was low where we have people. But the really phenomenal thing is that some of these uninhabited islands have coral cover, this is percent of the bottom that is occupied by corals, exceeding 80%, and this is unheard of. In fact, some of our colleagues didn't even believe that this could be true. And this isn't just one little location. This is an island-wide average where we dive every kilometer around the entire island, and we collect our data, and we have means and estimates of variability. But across those entire islands, we're seeing the bottom pretty much occupied only by coral. So despite all of the global impacts that are currently happening on reefs around the world with these warming and bleaching events, with ocean acidification, if you go to a reef that doesn't have pollution, fishing, sedimentation, the introduction of non-native species, you can still see reefs that on an island scale that are dominated by corals, that have fish populations that look like this. You jump in the water, you're immediately surrounded by sharks and snappers. And this is the Wild West. This is what reefs look like. This is what reefs looked like in the past. And this has been really important for serving as a baseline for us, because we're thinking about what the reefs of Hawaii looked when we first started scuba diving, and that's our baseline. But actually those reefs were probably way overfished even at that time. And so now to be able to say, well, this is what the potential for reefs is to produce a certain amount of fish. This is how much coral can actually live on the bottom if reefs are taken care of in a proper way. And so we've been trying to integrate this work with all of the human impact work to better manage these reefs. And actually, before President Bush left office, he created one of the largest protected areas in the US, the Pacific Remote Island Areas Marine National Monument. Many of you may have heard of this. It includes all of those unincorporated US territories across the Pacific, Rose Atoll, Howland and Baker, Jarvis, Kingman, Palmyra, Johnston, Wake. A lot of these islands were former military bases, or we grabbed them during the Guano Act back in the late 1800s. So these are all US territories. They're now protected. They're national marine monuments. We also have the Papahanaumokuakea National Marine Monument, which is the Northwest Hawaiian Islands. And actually just recently, President Obama decided or announced that he was going to create the world's largest protected marine reserve in the Pacific Ocean, which was basically a little bit cheating by just expanding those monuments that Bush had already made to include the EEZ, or the exclusive economic zone, around those islands. So I believe this was written into action recently, but not this entire area was included in that designation. So we're seeing, and actually in President Bush's designation, he used some text from one of our publications directly in the announcement that he was going to protect that area. And so that was the first time we realized that our science could actually reach the desk of the president and make a difference for protecting these islands. So we've seen definitely some positive successes with the work we've done in these remote areas, and we definitely believe we need to continue promoting this, especially just going back. Some of these reefs are part of the Southern Line Islands, which is part of the Republic of Kiribati, and they're the only islands in that uninhabited group that have this amount of coral cover, and they're still, as of yet, unprotected. And so we're going to be hopefully working with the Kiribati government to ensure that they're protected, because there's no people living there. So they're not providing subsistence fisheries for anybody. And they're literally just waiting out there for some longliner or Taiwanese fishing vessel to come through and take all the sharks off the reef and use them for shark fin soup. So hopefully we'll get those islands protected. So my goal today was to give you guys a taste for some of the research we do on human impacts, to show you some direct examples of how the research can be used for management, and to see some action from this science, and to share with you some of our work from the remote islands in the middle of the Pacific, to give you a taste for what coral reefs looked like 1,000 years ago, ultimately to help ensure that these ecosystems are around for future generations. So our kind of take-home message is we need to protect what remains, especially those healthy ecosystems in the middle of the Central Pacific, and we need to certainly do a better job at trying to restore what's been lost on these coral reefs across the world. So with that, I would like to thank all the members of my lab who provided a lot of the data in this presentation, and thank a variety of different funding sources, and then ask if there are any questions. So yeah, that's a good question. The question was about acidification causing damage to the reef and loss of herbivores also causing damage to the reef, but how might restoring herbivores might potentially positively reflect or deflect ocean acidification, yeah? So that brings up a good point. Much of what I talked about today was introducing you to these different problems, but in reality, they're all occurring simultaneously, and understanding how these different things interact. So how does removing of herbivores and ocean acidification together change the structure of these reefs? Certainly by bringing herbivores back, we're going to reduce the amount of all of these fleshy seaweeds which compete for space for with corals. So that's one thing. But if it just kind of depends on what time scale you're thinking of. If you're thinking about 200 years from now when the pH of the ocean is a lot lower than it is now, it's hard to predict, I guess, is the answer. Thinking about how these things interact is a lot more complicated, and we don't have a lot of data for these interactive effects, and that's kind of where the science is going right now. Yeah. Question was about how does elevated CO2 directly affect herbivore populations? There have been a lot of studies looking at ocean acidification effects on fish, and I don't think any have specifically looked at herbivores, but there have been a lot of studies on other kind of model fish species showing that fish lose a lot of their sensory abilities under ocean acidification. For example, fish can detect predators by a scent in the water, different chemical cues, and when you grow them under ocean acidification conditions, they actually swim towards the predator instead of swimming away. There are other kinds of neurological issues that we're seeing with fish under these ocean acidification experiments. So I don't know if herbivores are affected directly that same way, but yeah, there's a lot of interesting studies on that. Yeah. Yeah, that's a great question too, and I wish we could do it, but as of now, there hasn't been any successful effort at closing the life cycle of these fish in captivity. So a lot of the reef fish spend a fair amount of their time as little larval fish drifting in the open ocean, where they eventually get a cue to come back to the reef and metamorphose and turn into their little mini reef fish self. But that ability to metamorphose and to get them to settle out of the plankton in captivity has not been successful. And so there are efforts underway with sea urchins, which are also important grazers, and they're a lot easier to grow in captivity. And so I have seen there's been efforts in both the Hawaiian Islands and the Caribbean to grow out sea urchins and deploy them out on the reefs, but the reef fish are a harder one. We need stronger restrictions on fishing. In Hawaii, you can fish them without a fishing license. So, yeah.
Jen, thank you for a spectacular